Good afternoon, everyone. And on behalf of the Clinical and Translational Science Collaborative of Cleveland, we welcome you and thank you for joining us for our third READI Special Populations Roundtable talk, focusing on race and ethnicity in the context of ancestry and health disparities research. Before we begin, we'd like to share a bit about the Clinical and Translational Science Collaborative of Cleveland, or CTSC. The CTSC is a collaborative among Case Western Reserve University and its affiliated hospital systems, the Cleveland Clinic, Metro Health, University Hospitals, and the Louis Stokes Veterans Administration Medical Center. We aspire to be a catalyst for high-quality clinical and translational research, both locally and nationally, by changing the culture and environment of biomedical research, sharing resources and expertise, and streamlining the research process to move translational research from bench to bedside and to the community. Today, we have Dr. Rick Kittles. He was raised in Central Islip, New York. Dr. Kittles holds a BS degree in biology from the Rochester Institute of Technology, where he pledged Kappa Alpha Psi fraternity, and he has a PhD in biology from George Washington University in Washington, D.C. In 1990, he began his career as a teacher in several New York and Washington, D.C. area high schools. From approximately 1995 through 1999, he was a researcher with the New York African Burial Ground Project, a federally funded project in New York City, in which Howard University researchers, led by anthropologist Michael Blakely, exhumed the remains of 408 African Americans from an 18th century graveyard. Dr. Kittles gathered DNA samples from the remains and compared them with samples from a DNA database to determine from where in Africa the individuals buried in the graveyard had come. Beginning in 1998, as he was completing his PhD at George Washington University, Dr. Kittles was hired as an assistant professor of microbiology at Howard University in Washington, D.C., and also named director of the African American Hereditary Prostate Cancer Study Network at the university's National Human Genome Center. Dr. Kittles also co-directed the Molecular Genetics Unit of Howard University's National Human Genome Center, and he served in these positions until 2004. Beginning in 2004, he served as an associate professor in the Department of Molecular Virology, Immunology, and Medical Genetics at the Zagornis Medical Research Facility at The Ohio State University in Columbus, Ohio. He is currently the leader of the Washington, D.C.-based African Ancestry Incorporated, a genetic testing service for determining individuals' African ancestry, which he co-founded with Gina Page in February 2003. Dr. Kittles was featured in the BBC Two films Motherland, A Genetic Journey, and Motherland Moving On, and part 4 of the 2006 PBS series African American Lives, hosted by Henry Louis Gates. On October 7, 2007, he was featured on the American TV news magazine, 60 Minutes. In February 2008, he appeared in part 4 of African American Lives 2. He has published on genetic variation and prostate cancer genetics of African Americans. In addition, he discovered through a DNA analysis, he descends mainly of people of Dakar, Senegal, and Nigeria's Hausa people. Dr. Kittles is currently the Senior Vice President of Research at Morehouse School of Medicine. Welcome, Dr. Kittles, and the floor is yours for your presentation.
All right, thank you. I'm quite excited and honored to be here and talk about a little bit about my research and to share some insight, but also have some good discussions about how we leverage genetic ancestry in health disparities research. So let me pull my slides up here. Here we go. You guys see that?
Yes.
Okay. Do you see the full PowerPoint screen?
We see the presentation version with your notes.
Oh, with my note. Okay. I should have done this earlier. Let me see. How do I move this?
I think if you do another screen share.
Another screen share. Oh, here it is. Is that it?
No, if you go to the screen share, oh, here we go.
All right, my apologies. I will get this together. Let's try this again. Is that it?
This is it.
Okay. All right. So my title is Leveraging Genetic Ancestry in Health Disparities Research. And actually, this is the title of a paper that myself and some colleagues wrote that was published in the American Journal of Physical Anthropology several years ago. So it's well known that there's this paucity of genetic information in diverse populations, and that's been an issue for some time. This graph showing sort of the amount of non-European data in these genomic data sets is very low, and it has been for some time. It's just within the last maybe 5 years or so where things are starting to accumulate, but still nothing at the capacity that we see with European populations. And to be honest with you, this has slowed down sort of this whole effort of genomic discovery and production of precision medicine products for these diverse populations. One of the issues when we think about this whole issue of data, it's not just in the genomic realm, but it's also overall health. And for so long, there was just a focus on white men, and then there was efforts from the federal government to set up women's health initiatives and Black Women's Health Initiative and Hispanic Health Initiative. These initiatives help provide some useful information to move beyond just the model of white men. We also know that given the amount of genetic sequencing and research that's been done, for instance, around, let's say, cancer-related genes or diabetes-related genes, the bulk of that data being from people of European descent. And so minorities have a higher chance of getting what we call variants of unknown significance when you do these genetic tests for disease. In fact, African-American, Hispanic, and Asian populations report having a much higher number of these variants of unknown significance. What that means is when one sequences an area of the genome where there is known genetic variants that impact risk for disease or some outcome for disease, the bulk of that data is of European data that came from people of European descent. And so when we sequence those areas in non-European populations, we find these unknown variants. We still are trying to assess the impact of these variants. And it causes some frustration, not just with the researchers, but in particular among the men and women who are being tested. The level of stress and anxiety to go through a genetic test and then not be able to have a firm sort of decision based on those results. But also what's been problematic in the field has been the lack of robust methodology when one tries to explain human variation and global populations. Here in the United States, we've used these different models and they've changed, these definitions have changed through time. But when one talks about self-reported race, this issue of ethnicity and race, ethnicity being Hispanic, non-Hispanic, race being white, Black, Asian, what we're moving more towards is using ancestry as the better predictor, not predictor, but the better descriptor for these particular populations. Now, self-report is important, and I think it's going to continue to be a key feature in describing and categorizing populations. But ancestry is going to allow us to really look at the genetic background of these individuals and leverage that in our genetic work. And I'll give some examples of that shortly. When one looks at disease, complex disease in particular, there's a race, the social component. That's when we're talking about race. Race is a social political construct, and it's very difficult to tease apart the social from the genetic when you just use race. Let's say African American or Native American or Asian race. It's difficult to tease apart that genetic component. So now what we're doing is we're using race as this social construct in the sense of the environment, how they define themselves, but then genetic as a piece to describe the genetic background of these individuals. And then the complex disease due to the interaction of the two, the contributions and interaction of the two. I'm not going to get much into admixture mapping, but in the publication, we really talked about how you estimate ancestry and this issue of admixture and admixture mapping. But this goes back to the 50s. In fact, it was first proposed by an individual named Rife, who was looking at particular blood markers, proteins, and across different populations, and came up with these formulas for estimating ancestry. After that, Chakraborty and Weiss, and then there was a flurry of these theoretical papers that came out in the 90s and early 2000s that advanced various statistical strategies and methods. This is just a schematic showing haplotypes from source populations. Let's say the orange represents European, the blue represents African, West African populations. When you have that combined gene pool due to admixture, over time, those blocks of chromosomes break up into these chunks of shared ancestry. And that's what we leverage when we type ancestry in these individuals. We estimate overall the overall European proportion of European, West African, and Native American or Asian, if we see that. But we can also go marker by marker across these chromosomes and leverage that information in terms of predicting risk for disease or understanding particular drug treatment outcomes. So with these ancestry analysis, we have these global ancestry estimates. We have the estimates based on genome-wide data. And then they're useful, obviously, for association studies. You can control for confounding. You can look at these ancestral correlations with clinical measures and outcomes, and then also identifying incorrectly identified racial ethnic categorized individuals. So you may have a big consortium of, and you have samples and they may have been incorrectly categorized. Looking at ancestry allows you to really say, oh, this doesn't match with the self-reported feature. So then there are many ways to address it. Address confounding. You can do the principal components, dimensional reduction methods, and other methods using structure, and then the local ancestry inference, many different platforms and program software, and they don't differ that much, and they pretty robust in their estimates. This is just a schematic showing the spread of ancestry in Caribbean and neighboring populations. And I find this to be interesting because we see sort of the spread of European, Native American, and West African ancestry across these particular Central, South, and Caribbean populations, which represents sort of the spread of diversity. And so there's really no exclusive finite sort of groups, but a well-distributed spread of genetic variation and genetic ancestry across these groups. This is just a schematic showing local ancestry, because local ancestry is important too. On average, for early on, we were doing a lot of summarizing of genetic ancestry, and we would give you an estimate of genetic ancestry. But now we're actually looking at the local sort of marker by marker across these chromosomes to really see what input does ancestry have. So here on chromosome 19, we see a region where the ApoE gene is, and in various individuals, this is Puerto Ricans, these are African Americans, and these are non-Hispanic whites. You see the distribution of ancestry. So the European represents blue, the West African in orange, and you can see that for this particular region, Puerto Ricans and African Americans have a significant West African component compared to the non-Hispanic whites that were in their study. So we can look at local ancestry. We can also look at the global, what we call global ancestry, which is an estimate, for instance, of Puerto Ricans having 60% European, about 15% African, and then the rest Native American. This is another schematic showing the spread of ancestry across these chromosomes. And back in 2004, we worked with Mike Selden's group at UCSD, UC Davis. And we looked at across chromosome 5, about a 40 centimeter region, centimore region. And for the West Africans, you see here, European Americans and African Americans. And you'll notice that for that West Africans, it's pretty broad red West African component for that chunk of chromosome 5. And Europeans is blue. But for African Americans, it's a mosaic. And we see that throughout the genome for African Americans, this spread of both European and West African ancestry, depending on your family history. We've done a lot of admixture mapping using this methodology, and it allows us to really look at the average proportion of ancestry along all these chromosomes. And you'll notice that for the most part, for African Americans, it's about 20%. But then we have these deviations across these different chromosomes, and we can leverage that when we map genes for disparate diseases, like, for instance, sickle cell, or in this particular case, it was sickle cell modifying genes, but other genes that, like prostate cancer, using this method called admixture mapping. This is a schematic of my genome. This is my ancestry painting across the different chromosomes for me. And you'll notice that for the most, about 90% West African ancestry. The rest is European, about 10%, very little Native American. But you see here that for most chromosomes, I do have chunks of blue for European ancestry. I always like to show chromosome 15 because that's where the major genes for skin color are. And I don't have any European sort of alleles. It's all West African alleles for that chromosome. And those are the big predictors. That chromosome is the big the gene predictors for skin color. If I were lighter in complexion, I'm sure that I'd probably have a lot more European ancestry along some of those segments of chromosome 15. When we look at ancestry, our group was able to show that West African ancestry is associated with this increased burden of prostate cancer risk in African Americans. And we did this because we were looking at PSA and this whole controversy of prostate-specific antigen testing in the African American and European population. And what we did in this study was we looked at self-reported whites and blacks and looked at the predictability of PSA as a biomarker in these two groups. And this is based on self-report, and found that the prediction was better at the various cutoffs of PSA for African Americans than they were for European Americans, and which did provide some support to this whole controversy of maybe we should have race-specific cutoffs. But then we said, well, it has to be more than just this self-reported race. So why don't we just look at the African Americans and stratify them based on low, medium, and high West African ancestry? And in fact, when we looked at that, we saw that indeed there was this increase in predictability for PSA among African Americans that had higher proportions of West African ancestry. So this suggests that, yes, in fact, PSA behaves differently between whites and blacks, and a lot of it may be due to the fact that there's some genetic, due to the genetic background, there might be some genetic influence on the prediction for prostate cancer using the PSA biomarker. This is just a schematic showing the different allele frequencies for SNPs that were found. These are single nucleotide polymorphisms that impact prostate cancer risk. So when we look across the world, we find that for many European populations, the frequency of those risk alleles are low. Many Asian populations are very low, but the African populations, we see the higher frequency of these risk alleles for prostate cancer, which suggests, this evolutionary ancestral component to a disease that has a strong sort of disparity feature. This is a schematic showing the spread of variation for those SNPs that those prostate cancer SNPs in global populations, Africans, Europeans, Asians, and Native Americans. And about the first PC1 here just explains about 70% of the variation. And it separates Africans from the rest of the world. And so we see this feature throughout the genome for the most part, where we see sort of a different sort of background among what people of African descent and the rest of the world. And a lot of it has to do with humanity starting in Africa and then spreading out into Asia and the Middle East and Europe. So we looked at those variants for prostate cancer and found that with increasing risk allele dosage in Black men, we found increasing West African ancestry. So we saw this correlation between West African ancestry and these risk alleles for prostate cancer. Very, very fascinating, because like I said, what it suggests is that the genetic enrichment of these genetic alleles, the enrichment of these genetic risk markers in West Africa is probably what's driving a lot of the disparity that we see between Black and white men for prostate cancer. We also looked at genes for vitamin D and skin color, did a big GWAS, a genome-wide association study, and found signals for skin color. But we also were able to predict, using a genetic risk score, vitamin D deficiency, which plays a role in cancers such as prostate, breast, and colorectal cancer. And in fact, while it was the same genes that contributed to the risk for vitamin D, it was different alleles or different variants in those genes that actually contributed. And for African Americans, we were able to account for 76% of the percentage of folks who were vitamin D deficient having this particular risk score. When we look at other disparities, we don't necessarily see the same signal we see with prostate cancer, where there's a strong genetic component that accounts for the difference. For the most part, it's social, behavioral, social, cultural. When we look at head and neck cancer, it was really insurance status that was contributing to the disparity. We look at colorectal cancer, it may be dietary differences that we see impacting the disparity. So I think it's important that we recognize that the bulk of disparities have very little to do with genetics and more so with the social determinants of health. Lung cancer risks was an interesting finding, where we looked at West African ancestry and found that there was a difference, in particular among men, Black men, where we saw this increase in African ancestry among those with lung cancer and some of their clinical features. So this goes into precision medicine, because all of this plays a role. So as I mentioned before, paucity of data in non-European populations, the information that's garnered from just looking at Europeans has been driving a lot of the precision medicine. And so most of the information is currently based on populations of European descent. And besides that, we have these big economic barriers that actually may increase disparities, in addition to the data, the lack of data in diverse populations. And this is important, because what we're doing now is we're using precision medicine products to intervene in this disease process earlier, because the typical intervention is late. And when you have late intervention, then it's poor outcomes. So we're trying to intervene in this disease process earlier in time. I'm going to give you a quick example of one where we found that exploring genetic variation in African Americans is going to be useful for particular drug response data. So naltrexone, for many years, they knew it was effective in whites, but not Blacks for alcohol treatment, and then also smoking cessation. Here we see treated, untreated. This is the survival curve. We see a big difference there in the big difference there in outcomes between the treated and untreated. For African Americans, there was no difference here. So they did not respond to this particular drug for smoking cessation. This is the clinical trial that I'm talking about here. So what we did was we said, let's look at just the African Americans and see if, in fact, if we break them up based on African ancestry, if some of these individuals that have higher European ancestry, if they respond to the drug. So we typed the African Americans, looked at ancestry, categorized high versus low among the African Americans. And in fact, what we saw was those that had low West African ancestry. So these are African Americans who had a higher proportion of European ancestry. They actually responded. There was a treatment response that was significant between naltrexone and the placebo group versus those that had high West African ancestry. But when we looked at the whole sample pool together, we didn't see anything because it was the effect was hidden because of the because it was pooled and it was very heterogeneous. But this allowed us to say, okay, well, let's go in and see what genes are important in this response. And in fact, what we found was that the mu-opioid receptor, there's some variants there that are population specific, and in fact, drive the effect that we see in European people of European descent. One of the things I want to talk about is skin color and how it's used in the U.S. for it's classifies racially classified social groups and stratifies opportunity structures and life chances because of racism and systematic discrimination. However, skin color is also strongly correlated with genetic ancestry. So the study of ancestry and health outcomes may strongly entail both biological and sociological factors. So when we look at ancestry and try to understand disease, it's hard sometimes to tease apart the social component because ancestry and skin color are correlated, and skin color is a defining sort of feature in the social experience of people of color, in particular here in the United States. So anyway, I'm going to end with this. Some of the problems and pitfalls, marker selection, all ancestry markers are not the same. Local ancestry matters, IBD mapping methods are evolving, and we need to pay careful attention to make sure that any associations we see with genetic ancestry is not due to social cultural factors. And the use of pooled samples in genetic ancestry actually is synonymous with racial comparisons. And so we should perform the ancestry analysis within the self-reported group. So for instance, don't mix whites and blacks together in your study and use ancestry as a predictor, because it's very similar to just doing black-white comparison. If you look just within the African-American population, there's a spread of ancestry that's pretty broad, and it will allow you to really leverage that information. So here we see for African-Americans, the spread of your African ancestry is very broad, but for whites, the spread of African ancestry is very low, and there's not a lot of variance there. And so if you were to compare these two and together, it would be just like a black-white comparison because of those distributions. So that's one of the reasons why I'm very particular about when you do ancestry analyses for disease and outcomes, you do it within the self-reported groups. And I'll end with that. Thank you very much.
All right. Thank you, Dr. Kittles. Do we have any questions? You can place your questions in the chat and we can read them, or you can take yourself off mute and let Dr. Kittles know what's on your mind. This was a ton of really great, insightful information. So thank you so much. While people are thinking, I have a question.
Okay.
Where do you see the greatest opportunities for insights gleaned from ancestry and health disparities research in terms of advancing health equity?
I think the big sort of focus or area should be around precision medicine. And the example I gave you with the naltrexone, even though naltrexone is an old drug or whatever, we still see sort of the effect of ancestry on these particular treatment outcomes. And so I think that is an area where ancestry can really play a pivotal role, from going from the use of race, self-reported race, to actually looking at the actual genotypes of the individual, because that's what precision medicine should be about. And so understanding that there are some variants that impact drug response that vary across the globe, ancestry is quite helpful in identifying where they are and what's going on.
Excellent. Juliana, I apologize. I didn't see your hand. If you'd like to come off mute.
Hello? I didn't hear anything.
Okay. We'll give you a minute to come off mute, and maybe we can explore a little bit about the intersection of your work and the National Center for Advancing Translational Sciences. We are really focusing on solving for rare diseases now, bringing more treatments to all people faster. Through the science of translation, the process of turning observations from the lab, clinic, and community into interventions to improve health, scientists have learned that 80 to 85% of rare diseases are genetic. They aim to leverage what rare diseases have in common to make progress. Your focus has been on ancestry and prostate cancer in African-American men. Are there also genetic commonalities you found through that research that persist in other disease states in the African-American community?
So what's the question?
There is a lot in there, right? Do you see the same trends, if you will, in other disease states in the African-American community?
Yeah, so, these are disparities, right? But however, as I mentioned before, many of these disparities are not due to any genetic difference between Blacks and whites. They may be, let's say, let's say diabetes, right? Diabetes could be a genetic disease. I mean, it is a genetic disease, but there's a strong social component to that. And so when we try to tease apart the disparity, what's accounting for the difference between Blacks and whites in incidence and mortality from diabetes, it's not genetics. It's, social determinants, other social determinants of health. And so I think we have to be honest. We should look and see if genetics is accounting for that disparity. But if it's not, then we need to target particular behavioral interventions, like walking, a mile a day, or eating different, lifestyle differences, or even, looking at eliminating some of the structural racism, and that's contributing to a lot of this too. So what this is doing is highlighting sort of the need for holistic approaches to understanding disparities.
Okay, thank you. So let's say I'm doing research for the first time, but I'm interested in health disparities, health equity. What are some of the major considerations and precautions I should take as an investigator?
In terms of ancestry?
In terms of ancestry, yes.
Yeah, you should just be, it's ancestry, like race, is very contentious. And so you just have to be very careful in your presentation and your models and your design. So often people may not necessarily understand the history of race and racism, and the role ancestry plays in teasing apart race. And so people think, oh, well, we just group everybody together and just look at ancestry. That's all we need to do. Or they'll start defining ancestry very superficially, and it ends up hurting different communities. And so I would just say, be very vigilant in terms of the sensitivities of the communities that you're representing. And then also, I would suggest that that they justify whatever they did, the model that they used, so that folks can critique it. And if it's not a good model, maybe they can change it. Not maybe, but they should change it. But yeah, you should justify your models, and also be vigilant about the sensitivity of the communities that you're describing.
Okay. And in terms of being able to be sensitive and vigilant with the various communities and special populations. Are there any resources that you recommend that people who, again, we're just going to say, assume that everybody is new to this space? What are some films?
That's a great question, and I like how you put it. Assume everybody is new, because the bulk of biomedical researchers aren't trained in a lot of this, and so there should be community engagement courses and opportunities for many of these researchers so that they understand. It's hard for a lot of the researchers to even describe what health equity or health disparities is. I find it interesting. I was at a meeting once and there was a cancer center director and somebody asked him what's a health disparity, and he really couldn't answer. And he was a cancer center director of a major cancer center in this country. And so it was then I realized that everybody's not on the same level, the same page in this space. The training is important. And you can do that through workshops or whatever. I don't have a book that you can read. There's no book. Part of it is having a more robust training. A lot of my insight in this came from my anthropological training in graduate school. And because I had a strong anthropology focus. And so I think that also increased my sensitivity besides the fact of my lived experiences as an African-American male. So I think all of that together brought me to a different space than the person who trained in the lab next to me at NIH, I mean, we had different sort of upbringings and all of that. So, but now there's opportunities for engagement, programs like this, seminars like this, and symposiums where folks can sit and learn some new terminology and understand what these sensitivities are, what the critical issues are for the communities, and the need to protect them. We should look at what we do as providing insight and to increase the health of these communities.
Absolutely. And we do have 2 questions in the chat. Building on the cartoon, would you speak to the need to consider gender as well as ancestry? Yeah,
gender is critically important, and it has been for a long time. But it wasn't until NIH and Health and Human Services really started putting money behind research in women's health. I showed you the data on lung cancer, where we looked at ancestry, and we didn't see an effect in Black women. We saw the effect in Black men. And we see that, there's this, for many of these diseases, and many of these disparate diseases, there is this difference between men and women that we really don't understand, because we don't necessarily recruit large numbers of both of them to really explore those differences. Just looking at the impact of genetics, let's just say just in one gender, one could just imagine by looking at two genders that, the complexity is even more. Let me give you an example. So when one looks at modifiers of complex disease, many times they're looking just at men or women, but to be able to identify modifiers of disease, you need large sample numbers and all of that. Sometimes those modifiers differ between men and women, and which is why it's critically important to study both genders.
Thank you. The next question: self-identifying is often a more informal process in taking a medical history or screening for a clinical trial. If people are self-identifying when participating in studies, then how do we get the distinction between African-American and West African, Northern or Southern European, or any other distinction? Is the answer that genetic testing is going to be a required element for drug trials?
Yeah, I think it is for some already, and it's going to continue to be a requirement because of this issue of sometimes people don't know what their background, their family history is. They may say I'm African American, but their grandfather could be one of their grandparents could be of European or Native American descent. They may not know that, and so that would play a big role if 25% of your genome came from someplace other than West Africa, in terms of your response to a particular drug or, in a trial. You want to have as much information on those participants as possible. So self-report is important first stage, but genetic ancestry testing is crucial to really understanding the heterogeneity in those groups of self-reported groups, and then also the impact of genes in that maybe have an ancestral component to them on the outcome. So,
Thank you. Another question from the chat: where or how do you source your genetic information for your studies? Given the popularity of companies like Ancestry or 23andMe, within the context of historical systemic racism and disparities with regards to informed consent, Henrietta Lacks, I'm curious if you could speak to the modality of garnering your research volunteers and data so as to educate and inform the community at large.
I'm a little confused. So how do I don't know. What's the question again? What was the first part of that?
Where and how do you source your genetic information for your studies?
Okay. So yeah, I've collected data for my, I collected myself. And I do have collaborators that contribute at times, but for the most part, I've collected the data myself. And I started early on when I started my career at Howard University of engaging communities, consenting across different types of studies, whether it was alcohol abuse or prostate cancer or lung or colorectal cancer, and understanding how the community defined themselves. So that's why self-report for me is critical. And it got to the point where we would ask the participants, how would you define yourself? And they would say, African American or whatever. And then I'd say, well, what about your parents? How would you define them and where were they born, right? And then I would say your grandparents. And so that would give me, that would be 3 generations of data. And it would allow me to even assess, if they knew, what their genetic background was going to be. So if they say one of my parents is from Europe, one of my parents is from Europe and my other one is from, let's say, Kenya, I knew that 50% of their genome was going to be African. And so, in many cases, it correlated with those who knew their background, but most people didn't. And so that's why the ancestry was so important. In terms of the parental populations, the ones that we use to compare the source populations, a lot of that comes from genetic data sets. Also, my own recruiting of samples in West Africa. But what was critical was as I was doing this, it was all centered around different studies. And so I would consent for those studies. And there was no sort of opportunities. Not no opportunities, but there was no interest in selling that data or not doing anything that I wasn't supposed to do with the data based on the consent. So I know that for many communities, there is this fear of exploitation and being experimented on, and the whole Henrietta Lacks story. But a lot of that is minimized now because of human subjects protocols.
Okay, thank you, Dr. Kittles. Any other questions, comments, concerns from the group? If not, we thank you again, Dr. Kittles, for all of your insights. Your research is incredible, and we can only aspire to continue learning from the ground floor and building up and beyond as we learn more about health disparities, health equity, and really advance towards health equity. So next up, we have our READI Special Populations Roundtable talk about geography. We'll be looking to learn more about rural communities. We'll focus on Cuyahoga and Summit counties with Dr. Prakash Ganesh, Medical Director of the Cuyahoga County Board of Health. What can you do today? You can schedule a research equity, accessibility, diversity, and inclusion consultation at spark.case.edu. And we are more than happy to help you explore, identify opportunities, and really just dive deep into health equity and health disparities research. You can sign up to attend future programming and join our listserv. And you can also visit our website and review some CTSC DEIA resources. We're going to give you a couple of minutes for virtual commute time. Engage in meaningful activity that will help you commute back to yourself, move, breathe, engage your senses. And that's from Dr. Jennifer A. King. Thank you so much for joining us. Feel free to connect, send us an email. We are really grateful for your time and investment, and we hope you have a great afternoon. Thank you.